back, everyone. In our last video, we talked about the differences between haploid and diploid and what that means for meiosis. So now what we're going to do is we're going to dive deeper into the actual phases of meiosis. So meiosis is the process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes. Again, so the copy that you get from mom and dad, okay, in our bodies is a total of 46. What we're trying to do is we're trying to separate those. Okay, we're going to unpack what that looks like as we go through this thing. But meiosis essentially is mitosis that happens twice. Okay, and it's really easy to remember how we label them. The first division is called meiosis 1, and the second is called meiosis 2. So again, it's basically mitosis happening twice with a couple of extra twists. No pun intended. So by the end of meiosis 2, the one diploid cell becomes 4 haploid cells, all genetically different. So the key phrase here is that one diploid cell, or in our bodies, one cell that has 46 chromosomes, is going to reduce and become 4 haploid cells, or 4 cells with 23 chromosomes. So the half the amount of information. Okay? And the key idea here, they're all genetically different. In other words, no sperm and no egg is genetically identical unless you are an identical twin. Okay? So if you look at this picture, okay, you're probably thinking in this picture right here, oh, there's some interesting stuff going on there. Okay, we're going to talk about that in a little bit. But we are going to separate the homologs. Okay, so as you can see here, in meiosis 1, we end up with 2 cells, okay, with already half the amount of information. So if you were to go through, okay, at the beginning, we start with 4 chromosomes. If you already look at the end of meiosis 1, we have 2 chromosomes here and 2 chromosomes here. Okay, and remember, we're trying to separate the homologous pair. Okay, these two are homologs. Well, the blue one went this way, the orange one went this way, so we separated them. And again, in meiosis 2, we don't want sister chromatids, we want our DNA in singular chromosome form. So we're going to reduce down one more time. Okay, notice how none of these cells are the exact same. Okay, every single one of these cells is different. And these are haploid cells because they have half the amount of information than what we started with. Right? Over here we have 4 chromosomes. Over here we have 2, 2, 2, and 2. So they're all completely different. So starting with meiosis 1. So just some background information before we kickstart this thing. Okay? Just prior to meiosis 1, the cell undergoes a round of chromosome replication called interphase 1. In other words, this is the exact same as what's going on in mitosis. We have interphase 1 before that body cell heads into prophase 1. Okay, so each replicated chromosome consists of 2 identical chromatids joined at the center. You already know that information. Okay, but the goal of meiosis 1, you have to write this down. We are separating homologous chromosomes. We're going to separate those pairs. So if you look, this is a synopsis of meiosis 1. Okay, there's not a lot that you can really see in here. But again, we're trying to separate these 2 pairs. So if you look really closely, we have a blue and a red one, blue and a red one. We are separating those apart. So we have a red going this way, a blue going this way, and so on and so forth. And those 2 cells produced at the end are not the same. Okay, they're genetically different. So prophase 1, there's a lot that happens in prophase 1. This is one of the most important phases of meiosis, just as a whole, and you'll see why. Okay, so the first step is that those homologous pairs, those chromosomes, are going to pair up to form what is known as a tetrad. Okay, and if you're good with your prefixes, tetra means 4. So in other words, we are going to have 4 chromatids lined up against each other. Okay, I have a picture that's going to help you understand this in just a second. As homologous chromosomes pair up and form those tetrads, they undergo a process called crossing over. Okay, these 2 things right here are going to lead in an increase in genetic variation. Okay, it's very, very important. So when crossover happens, what it means is that chromatids of the homologous chromosomes are going to swap genes. Okay, they're actually going to switch DNA with each other. It's very interesting and quite fascinating. But again, what this leads to is it increases genetic variation. So if you look here, okay, imagine that these two are stacked on top of each other. And if you were confused by what I said before, we have chromatids here. So 1, 2, 3, and 4. Okay? And so as they cross over, you'll notice that afterwards, okay, we swapped, looks like the bottom right of this information on the blue with red. And then on the left side here, we swapped. And so now we have a little bit of that blue information on this red chromosome. Okay, this is the process known as crossover. Okay, and what this is going to lead to is it's going to lead to genetic information, and you'll see what that looks like at the very end of meiosis. So metaphase 1. Back in mitosis, I told you to make sure you know that metaphase means what? Middle. Okay, that's the same thing. So during metaphase 1 of meiosis, paired homologous chromosomes line up across the center of the cell. So in other words, it's the exact same thing, okay? Metaphase middle. So the key thing to remember here is that those homologous chromosomes are lined up double file, okay? So here's a helpful picture for you. So let's say that red chromosomes come from dad, blue chromosomes come from mom. They are lined up double file, okay? So there's one here, one here, one here, one here, one here, and one here, okay? We're going to separate these. As you can see, that dotted line is saying, hey, this is where we're going to separate. This is the middle of the cell. And so we're going to pull these apart eventually. Okay, but it's the same thing, right? These centrioles all the way over here, okay, on opposite ends of the cell, we call them poles, okay? They're going to release those spindle fibers and they're going to grab onto those centromeres. Okay, again, this is a lot of vocabulary, so make sure you understand your vocabulary. The centromere is the center of those sister chromatids or those chromosomes in there, okay? And that's what I call those hands that spindle fibers are the hands that reach on and grab onto the centromere to eventually pull them apart, okay? And we know that anaphase, right? Same as mitosis, we said anaphase apart, okay? So if you're looking, okay, we're just pulling those apart. So again, we're separating those homologous chromosomes. They move to the opposite poles of the cell is what this picture over here says, okay? So again, anaphase apart. Make sure you remember that. Make sure you lock that in so that you know exactly what happens every single time you hear the word anaphase, okay? Jumping into telophase 1 and cytokinesis. So very similar to mitosis, during telophase 1, a nuclear membrane is going to form around each of those clusters of chromosomes, okay? So notice the chromosomes. Are they the same in those cells? They're absolutely not, right? We have a red chromosome here with a little bit of blue. Here's its homologue, okay? It's the same size. It's got different information. Again, here's another smaller red chromosome. Here's its homologue in the middle. This one's blue, but it's with red. Okay, so these two cells that are already being produced are not only half the information than what we started with, but they're also completely different. Okay, and so just like we say in mitosis, cytokinesis is going to form those two new cells, right? And we have that cleavage furrow, the pinching as it's labeled here, so that the cell knows exactly where to cut and to divide completely. But again, half the information than what we started with, okay? So jumping into meiosis 2, okay? What meiosis 2 is we are going to basically do the exact same thing because we have sister chromatids. Well, in those daughter cells, we don't want sister chromatids. We want singular chromosomes. So that's why we go through another phase of meiosis, otherwise known as meiosis 2, okay? So unlike the first division, neither cell goes through a round of chromosome replication before entering meiosis 2. You might be wondering, what the heck does that mean? It means that there's no interphase between meiosis 1 and meiosis 2. We skip over interphase, okay? So interphase only happens at the very beginning of meiosis 1, okay? And so you might be asking, what's our goal for meiosis 2? We are trying to separate sister chromatids, okay? So meiosis 1, we're separating homologs or homologous chromosomes. In meiosis 2, we are separating those sister chromatids. Okay, here's a synopsis of what we're going to do. So prophase 2. As the cells enter prophase 2, their chromosomes, each consisting of 2 chromatids, become visible. So the chromosomes do not form tetrads because their homologous pairs are already separated during meiosis 1. So again, all we're going to do is those new spindle fibers are going to start to grab onto the centromeres once we line up down the middle in metaphase. But the big thing here, centrioles are going to move to the opposite poles. Okay, metaphase, what do you think this one means? You bet, metaphase middle. Okay, during metaphase 2 of meiosis, chromosomes line up down the center of each cell. Okay, another thing to write down here is that during metaphase 2, they line up single file. Okay, remember in my metaphase 1, we line up double file. In metaphase 2, we line up single file. Okay, anaphase 2, you bet, anaphase apart. This is easy. We're just going to split. Okay, so as the cell enters anaphase 2, the pairs of chromatids are going to separate. We're going to pull them apart to opposite ends. Again, the centrioles, releasing those hands or those spindle fibers, are going to grab onto the centromere, pull them apart. Okay, so telophase 2, as well as cytokinesis. So again, we're going to have that nucleus start to reform around those new cells that are produced at the end. Okay, and so these 4 daughter cells now contain the haploid number. Okay, so at the beginning of this slideshow, we started with 6 chromosomes in the middle. Well, if you look really closely, we have 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1, 2, 3. So each one of these has different amounts of information. Excuse me, they have different chromosome colors. They're all genetically different, but the number of chromosomes is the exact same. So 3, 3, 3, and 3. So the haploid number in here would be 3. In our human gametes, they are 23. Okay, we go from 46 to 23. Okay, so here's a synopsis of meiosis 1. And here's a synopsis of meiosis 2. So in the slideshow, I actually have linked to Amoeba Sisters for meiosis 1 as well as meiosis 2. If you need to go back and watch those, please feel free to watch those. I hope this was helpful. Thank you.